Okay, welcome everybody. And thanks to the Geological Oceanography Lab for organizing the food and all this vegetarian stuff.
Vegetarian stuff.
No, it was a joke.
Okay.
So I'm very happy, excited to introduce Adina Paytan today. So I'm going to give you a little introduction on her background.
Look, he's got a whole notebook. Be careful.
Yeah, actually I have more notes, so it's going to be a half an hour, 2 hours. So, but I just want to give you a little idea of where she's coming from. So Adina has obtained a bachelor degree, a double major in biology and geology, perfect for most landing, at the Hebrew University in Jerusalem. Then she moved to obtain 2 master's degrees, one in science education at the Weizmann Institute of Science in Rehovot. And a master's degree, that was pretty good, in oceanography at the Hebrew University. So then in '89, she moved to San Diego with Ron, her husband, and started a PhD at Scripps with Miriam, right? Miriam Kastner, on the marine barite cycle. So she's a marine biogeochemist, by the way. Forgot to mention that. Not a physical oceanographer, in case you're wondering. Then she did a postdoc at UCSD, San Diego, on a high resolution, to build a high resolution sulfur isotope curve for the past 120 million years. So high resolution for 120 million years is kind of an interesting combination of words. In 1999, she moved to Stanford, where she was a professor in the Department of Biological and Environmental Science for 8 years, I believe. And finally, in 2007, she moved to UC Santa Cruz in the Institute of Marine Science as a research scientist. So she has an impressive amount of well, first of all, the honors and awards. There's an incredible list of awards that I can't read today because it's really very long. And incredible research activity with a very large amount of funding and a very prolific number of publications, about 150 in peer-reviewed journals. And I start counting them, and she said, stop, don't count them. But really a lot of interesting paper in very highly ranked journals. So she's very prolific, and she has been working on very different topics within the overall marine biogeochemistry work. So today she will be presenting data on the impacts of ocean acidification on coral reefs, perspectives from a field study in Mexico. Thank you, Adina.
Thank you, Ivano, and thank you for inviting me. You turn the lights off near the...
Turn off the lights.
Yeah, just near the thing, so... Perfect. Can you all hear me pretty well? You don't need this thing. Move it out of my way, because I like to move when I talk. So thanks all for coming here and spending your happy hour on a Thursday afternoon listening to some science over here. And as Ivano said, this is just one of the projects that is ongoing in my lab, but I thought that that might be of particular interest to some of the people over here. So we decided to talk about ocean acidification. And just a brief outline of what I'm going to talk about today, because I am not sure how much background you have on the topic. I'm going to spend a little time describing what is ocean acidification, how does it work, and why do we care about it. I'll mention a few research directions in the community, not necessarily in my lab, to address ocean acidification, and then I'll spend most of the time talking about my research or my group's research, which brings me back to my first slide. This is a collaborative project with students in my lab, as well as other faculty at UC Santa Cruz, researchers in Mexico, and Ann Cohen from Woods Hole. Everybody contribute a little bit to this whole project because this is, in my opinion, one way of being creative and moving forward in science is to work with other people. This is also keeps me engaged in what I'm doing, because if I had to do the same thing over and over, I will be bored to death. So that gives me an opportunity to learn from my colleagues. Okay, so let's start over here. How many people in this room have not seen this curve before? And I hope I would not see anyone raising their hand. Good. So, whew. Okay, so this is, I'm sure everybody has seen that. This is the concentration of CO2 in the atmosphere over time as measured in Mauna Loa. It's called the Keeling Curve. And you can see that we have CO2 in the atmosphere has been increasing over the past decades. This is attributed primarily to fossil fuel burning and adding CO2 to the atmosphere. What are the consequences of CO2 additions to the atmosphere? Anybody can tell me what that causes? Come on. Global warming, yeah. Global warming, sea level rise, melting of ice caps in the polar regions. And was actually, global warming was the major focus of research that related to atmospheric CO2 until recently, primarily because the scientific community was not too worried about the other CO2 problem, which is CO2 dissolving in seawater and changing seawater chemistry. To a certain degree, the reason for that was because there's a lot more carbon in seawater and ocean has a buffering capacity, and we thought that that's not going to be a major problem. We were wrong. And as you can see here, what I'm talking about is the CO2 that dissolves in the ocean. This purple line over here is the same curve of CO2 in the atmosphere in Mauna Loa. The blue curve, is a curve of what is thought to be in, or what should have been in the atmosphere based on records of how many barrels of fossil fuel are being used worldwide. So we know how much fossil fuel we're burning. Sorry. This is what we should see in the atmosphere based on these records, which are pretty good records. This is what we actually measure. So you can see there's a pretty large discrepancy here. About 30% or so is missing. Where is all this missing CO2 going? Some of it maybe is going into new growth of trees and forests on land. Turns out that actually overall there's more deforestation than new forest growth, so that can't really account for this thing. Most of it is actually dissolving in the ocean. This is not something that is debatable or questionable or surprising. If any of you took any class in chemical oceanography, you know that all gases in the atmosphere, in any atmosphere, are in equilibrium with the water that they interact with. That's Henry's law. It really, the amount that you'll find in solution depends on the concentration in the gas phase, plus temperature, salinity, and other variables. So that's something that any chemist will agree with. It's the basic principles of gas dissolution. So if we have more CO2 in the atmosphere, we'll have more CO2 in the solutions or in the ocean. How does that work? You can see over here, CO2 is dissolving from the atmosphere into the ocean, increasing CO2 dissolved in seawater. Now, CO2 is a very special gas in seawater. It reacts with water to form carbonic acid. Carbonic acid then dissociates to form bicarbonate, releasing a proton here and increasing the acidity. Some of these protons then react with carbonate ion concentrations to form more bicarbonate. The end result of all of this process is that acidity increases, or the amount of the number of protons, the concentrations of these hydrogen ions over here increases, and carbonate ion concentrations decrease. Bicarbonate increases slightly and carbonic acid increases, but these two other effects are more important because the relative change is larger. How do we know that this happens? If you look at this figure down here, again, the red line is our Mauna Loa curve, CO2 in the atmosphere. Luckily, we have some monitoring programs ongoing, and this is the dissolved CO2 in seawater at the hot monitoring station off Hawaii. And you can see it increases in parallel to the CO2 in the atmosphere, and at the same time, in the same station, the pH of the water decreases as we expect. We can find that this anthropogenic CO2 has penetrated the ocean to a depth of about 1,500 meters in some places. This curve shows you how much anthropogenic CO2 is in the ocean on a transect from south to north. And we know that from the carbon isotopic signatures of the CO2 that is in the water. More than 500 billion tons of CO2 emissions are now in the ocean. So what are the consequences of that? Some modeling results show here the difference or the change in sea surface pH that resulted from this CO2 absorption onto the ocean. This is the difference between the pH of the ocean at 1700 and 1990. And if there was no difference, you'd see the ocean in this white color. All of these other colors show a negative value, which means a decrease in pH. And you can see that everywhere in the surface ocean, the pH has been decreasing. And you look at these numbers and you may wonder, well, that's a really small number. Minus 0.1 units of pH. Is that significant or not? It looks small. I just want to remind you that pH is reported on a log scale. Therefore, a 0.1 unit is actually a pretty large difference in pH, about 30% change in pH for 0.1 pH unit. So that's a large difference. So what does that mean? If you look again at this map, it shows the same data with now projection to the future. Of course, these are based on models, so it depends on what emission scenario we use. But in general, you can see that while in 1875, most of the ocean pH was around 8.2, 8.3, 1995, it's still above 8, 8.1, 8.2. 2050, you can see that it starts dropping below 8, 7, or below even 7.9. And by 2100, it will be 7.8 or 7.7. So that's a very big pH change in the ocean. What are the consequences of these pH change? So you notice that I put on the title here, potential impacts. I wrote potential because we don't actually know for sure, other than we are confident that the pH will decrease because already there's more CO2 in the atmosphere that will equilibrate in the ocean. What impact that will have on organisms is debatable, but these are the potential consequences. It might change the calcification rate of organisms that form shells. There might be shifts in nutrient and trace metal speciation and availability, in phytoplankton diversity, in growth and reproduction rates, tolerance to environmental fluctuations or other stressors, and again, a whole range of other potential impacts, food webs, all of which may have also influence on the people that rely on the ocean for their fishing, recreation, and so on. So there's lots of potential impacts, and as a result, various national and international agencies came out with a call to the community that we need to a fully integrated program of laboratory, mesocosm, field monitoring, modeling approaches to inform, to provide policy makers and inform management about strategies to address how humans might best mitigate or adapt to these long-term changes. So there's a lot to be done, but as a result, funding agencies had special calls to focus on ocean acidification, and all of these various aspects and programs were encouraged, and a whole array of community of people. I don't think I have that figure, unfortunately, but in other talks I show a figure of the increase in publications that have to do with ocean acidification versus time, and it looks exactly like the climate change hockey stick. It's an exponential increase in the number of papers on ocean acidification with time at a rate that rivals the CO2 increase in the atmosphere, actually. So that's actually a really good thing. We're learning a lot about ocean acidification and the consequences. What I'm going to focus about in my talk specifically is the impact of ocean acidification on marine calcification. And when we talk about calcification, we're now concerned or want to focus on the availability of carbonate ion concentrations. Carbonate is used by calcifying organisms to form their calcium carbonate shells. If there's less carbonate in the ocean, you would assume that it's going to be harder for these calcifying organisms to form their shells. And if you look over here, this is the same kind of map showing you the difference in seawater, surface seawater carbonate ion concentrations between 1700 and 1990. And you can see again that all of the numbers are negative with a significant decrease. The units are millimoles per meter cube of water, and you can see a decrease in the amount of carbonate ions available for calcifying organisms. When we talk about calcifying organisms, it's not just one or two groups of organisms. These are all of the different groups, phyla, organisms that some parts of their body involves the need for calcium carbonate. You can see they range from coralline algae, corals, bryozoans, other mollusks and echinoderms and so on. And a lot of these are either at the base of the food chain, so if they disappear or are not going to be abundant, that will have impact on fisheries. Others, like corals, have other important values to humans, so that's a really important thing to study calcification. Again, I'm going to zoom in a little bit further and focus on calcification by corals or coral reefs. And again, corals are relatively simple organisms. They're very skinny, or they're made out of 3 layers of cells. They're a sac made of 3 layers of cells, so they're exposed to the chemistry of the water around them. They form their carbonate skeletons from a mineral called aragonite, which is even more soluble than calcite. So they might be more sensitive to changes in carbonate concentrations. And in addition, they're already impacted by a whole array of other stressors: pollution, overfishing, people trampling around in the coral reefs and walking on them, and so on. So they might be already a little bit stressed. Of course, warming has been shown to impact negatively corals. So if you add yet another stressor and remove their carbonate ions, which they need to calcify, they might potentially hit a tipping point and will disappear. If you look at this map over here, again, this shows you now the aragonite saturation. Aragonite saturation is the concentration of calcium multiplied, or the activity of calcium multiplied by that of carbonate ions, because calcium on the timescales we're talking about remains relatively constant in the ocean. Basically, it shows this change depends on the carbonate ion concentrations. Again, 1765, everything, all the little black dots here are places where we have corals. You can see that the corals are bathing in waters that have a carbonate, aragonite saturation about 4.5 to 5. This is the optimal range for calcification for these corals. If we go to 1995, you can see there's still some red areas where most of the corals are, but the rest of the ocean goes to about 3.5 to 4, slightly lower aragonite saturation. 2040, 2100. At 2100, most of the corals are bathed in water that have a saturation level less than 2.5. At 2.5, it's marginal to low conditions for their calcification. These marginal, low, adequate, that's based on mostly on a lab experiments where you take a coral and grow it under controlled conditions, and you see a drop in their calcification rate once saturation goes below about 2.5. So based on these results and predictions, this coral demise got a lot of media coverage. The ocean's acidity is killing the corals. The Great Barrier Reef is going to be hit, and so on. And a lot of concern in the community about what's going to happen with coral reefs, what's going to happen to us, people who like to dive and look at these beautiful corals, or people that depend on the corals and coral reefs for their life. Again, a similar thing showing that by 2050, probably there's not going to be any more corals living on the face of this planet. I want to remind everybody that, again, these predictions are based on the assumption that once the saturation level with respect to aragonite falls to about 2.5 or lower, the corals are going to not be able to calcify and will disappear and not survive anymore. And that, the prediction of this value of 2.5 is based on what we expect CO2 in the future to be, and on lab experiments where corals were grown under these conditions for a short period of time in the lab outside of their natural environment. When you look at these experiments, this is a really nice paper that I like a lot by Fine and Chernov. They took a coral from the Red Sea, put it in an aquarium, circulated low pH water that has been equilibrated with high PCO2, and let it grow for a few weeks. And you can see that this coral ended up being naked polyps. So the coral actually did not die. It grew pretty nicely. It grew pretty fast and well. It photosynthesized. Their symbionts were there, but it stopped calcifying. So maybe corals can actually survive this reduced ocean acidification or reduced saturation level, but the coral reefs may not. That still is a problem because the coral reefs are what we call the rainforests of the ocean. They're an area of or ecosystem with a lot of biodiversity. They're nurseries for a lot of juvenile fish. And these are the structures that protect the coasts from lots of storms and hurricanes and surges. So if the coral reefs will disappear, that's also not a good thing. We also don't know if these sea anemone type corals or polyps, single polyps, are actually in the natural ecosystems are going to be able to survive. Maybe they will be more prone to disease or to other predators that will get them. Having said that, this is actually a pretty interesting, from a geological perspective, a pretty interesting observation, because this can explain a lot of the puzzles that geologists had with the geological record of fossil corals. If you look at the geological records, you see certain coral species abundant, and then they disappear from the record, and a couple million years later, they appear again, and the same fossils show up. So if they got extinct, it's really very, very unlikely that evolution will repeat itself to produce the same kind of organism. It's more likely that they either did not calcify but maintained or survived through that low period, or that they were few and far beyond secluded, and then when conditions turned back to favorable conditions, they became more abundant. But that was actually a really nice additional information from this specific study. Okay, this shows you again another lab work where you take the corals and you expose them to different increasing CO2, and you can see over here it doesn't matter if you change the saturation by adding HCl or by pumping CO2 by about saturation of about 2.5 down here. You can see a sharp decrease in their calcification rate. And if you take a whole number of different studies, they're all pretty much consistent that as saturation decreases, calcification rate decreases. The absolute numbers vary from species to species and from system to system. That's why they normalized it here to the pre-industrial conditions of that specific coral. However, when you look closely at the literature, you can find some unexpected results. So this is a study that shows that here that's normal PCO2 and high PCO2 at 25 degrees. There's actually no difference in their calcification rate of that specific coral under the gross or the experimental conditions in that lab. But when you change the temperature and you increase the temperature, that's when you start seeing a decrease in calcification. So it just tells us that things are somewhat more complicated than that simple one-to-one. CO2 increases, carbonate saturation decreases, you hit 2.5, the corals stop functioning. It shows you that different corals may respond differently and that other parameters may have confounding or building up or effects that interfere with each other. This is another study where they took a core and went down the core, and you could see that the pH over here reconstructed based on boron isotopes fluctuates quite a bit naturally from about 7.9 to 8.2, but then if you just smooth this variability, you don't really see any trend towards a significant decrease over time. And again, the reason for that and why is a big debate when you're looking at experiments in the field or when you're reconstructing experiments where you don't have other variables that you monitor in parallel. Basically, what I'm saying is that as more studies came up, it turned out to be that we don't know enough about whether or not these corals are or are not going to disappear and how they will disappear. And the main question that remains is whether the corals generally or certain species of corals have the ability to acclimate or to adapt to these changing conditions. If they do, then maybe they might, if they are protected, they might be able to survive these changes, which is somewhat good news. So in order to answer this question, you can't really take a coral out of its natural system and put it for 2 weeks or 2 months in an aquarium or in a beaker in your lab. For various reasons. A, the time span of that experiment is too short to enable adaptation or acclimation to happen and for you to monitor it. And B, you're removing the organisms from its natural environment where all of the complexity of the conditions in nature are removed. I'm not saying that you should not do such experiments. You should definitely do such controlled experiments where you monitor the conditions and look at the responses. But that's not enough. And you want to look at the response of organisms also at the ecosystem scale, meaning in their natural environment with all of the interactions in nature and for longer periods of times where they have or had the chance to acclimate and adapt. And one of the first studies to do that for coral reefs was published in 2011, and they looked at a coral reef environment in Papua New Guinea where there's natural volcanic CO2 springs. And they compared areas where the pH was on average 8.1 and they had a healthy reef system to an area close by where the pH was 7.7 because of these CO2 volcanic springs. They compared a whole range of different parameters over here, and you can see that a lot of them decline. So one is the relative change in this environment compared to the control. If it's lower than 1, it's the percent change. So percent hard coral cover, hard coral richness, and so on. These all decreased. However, if you look at the stuff that is on the plus side or increased, you can see that non-calcareous algae and seagrass cover both increased. Not very surprising. These are algae. They need CO2, so they get more CO2 over here, and they can do pretty well. However, this over here is massive porites. You can see over here these coral heads are massive porites right next to these CO2 springs at a pH of 7.7. So some corals seem to be able to grow or survive under these low pH conditions in the field. One of the questions are, how come these specific porites, coral heads, massive porites can do that? Or whether they were there before the spring and they're just maintaining. This is another potential explanation, because these springs they come and go. Their lifespan is relatively short on the scale of several years, and then they pop out. Another area of pressure builds, and the springs pop up in another place. So that actually is something to follow up. And one of the, as I said, the criticisms of this specific volcanic system is that the volcanic CO2 springs have too short of a lifespan to be able to address some of these questions. That's where my work comes, and I work off about 20 minutes south of Cancun in the Yucatan Peninsula in Mexico, near the Great Barrier Reef, a wonderful field site to work in. And this system is very different. The Yucatan Peninsula is a carbonate karst platform. What it means is that as the rain falls, instead of forming big rivers that drain into the ocean, the rain dissolves the carbonate and forms underground rivers, or caverns and cenote systems. And then these underground rivers discharge still in the ocean, but at submarine springs. Right off the coast over here, near a place called Puerto Morelos, there's about 15, 16 of these submarine springs. The locals call them ojos, which is eyes, because they're very focused springs discharge sites. You can see it over here. It's along some kind of fault, and these springs discharge water that used to be rainwater. It's actually groundwater. It passes through the carbonate, so it dissolves some carbonate. It has high alkalinity, but it also has a lot of CO2 in it from respiration in the soils along the pathway of the groundwater, and it has high CO2. So you have these low pH springs discharging in the coast, right at the Mesoamerican reefs, so it's a nice site. Advantages over the CO2 volcanoes, these systems have been operating and have their geological features. They're not ephemeral. They've been there at least since the last deglaciation. So there's many, many thousands of years of these caverns that have been operating and discharging water. The disadvantage is that this is presumably fresh groundwater. Luckily, in this system, there's mixing between seawater and the groundwater before the discharge. So seawater intrusion mixes with the groundwater, and the water that discharges is brackish water. Still has the high CO2, but the salinity is only a little bit lower than the surrounding seawater. If you look at the system, these are just the different ojos up and down the coast. This is how it looks like from when you dive over there. It's very shallow, 5-6 meters. And that's like Ojo, how it looks in a calm day. This is all of the resorts on the coast. It's about 500 meters offshore at a depth of 5-6 meters. And you can see the buoyancy over here of the waters that are bubbling out of these springs. You can see that they're very small, meaning that if you take a sample here and the sample about 2 meters away, conditions other than the discharge are identical. So light penetration and everything else is the same. It's the same coral system, but you've got a control site and an Ojo site very close to each other. The first thing you need to do to convince yourself and everybody else that that's actually something you can work with is put sensors to measure the chemistry conditions of these springs. To ensure that really they're discharging continuously, that the pH is always low, that you can calculate the saturation, and so on. So we deploy these sensors, and you can see here there's a lot of variability, which is completely not surprising. When you have springs, the discharge of groundwater depends on the tides. At high tide, there's less discharge. At low tide, there's more discharge. Simple hydrogeology because of the change in the head. So you've got more and less discharge based on tides. The other thing, it's a very shallow seagrass area, so there is diurnal cycles between photosynthesis and respiration that impact the pH of the water as well. And of course, they're seasonal. If you have a big storm, you've got lots of rain, you've got lots of discharge. So lots of variability. Despite this variability, you can see over here that with the exception of these are samples that were taken every hour. So with the exception of 1, 2, 3, 4 tiny periods of time, the pH was below 7.8, more like below 7.6. So continuously, these springs are discharging low pH water. You can see over here, again, different days. We also measure the calcium concentration here because as in contrast to the open ocean where the calcium doesn't change because the spring does dissolve, or these freshwaters go through carbonate rocks, they dissolve some of the carbonate. Calcium can be higher than seawater, so you have to monitor calcium in order to use that when you calculate the saturation level. And we also measure total alkalinity and DIC in the system, so we have a full control on the carbonate chemistry and other chemical conditions of the waters. The second thing we did is we went ahead and tried to look at what lives near these springs relative to the control site. We laid transects along, a distance, so we had 0 to 2 meters, 12 meters from an Ojo center or along a fault line where you have discharge, and we pretty much recorded everything that grows and lives within these transects and compared the control sites to the Ojo site. That's the results of these comparisons. This is not an easy figure to read, so I'll walk you through that. The numbers on the top here, so each one of these 3 panels is a different spring or a different Ojo. The numbers on the top are just meters along our transect. So we started a transect over here, that's 0 meters. That was a 12-meter long transect over here. This one was a 9-meter long transect. This is 3 meters that you can see over here. The colors below the numbers are the aragonite saturation levels based on our discrete sample collections and our monitors, our sensors. So we looked at the DIC, alkalinity, temperature, salinity, calcium, and so on, and calculated the aragonite saturation. The red spots over here at the edges of these transects is a aragonite saturation of about 4.2. That's your average background in this lagoon. You can see it over here, here, and so on. So at 0 meter, you have a saturation of 4.2. At 4 meters, so just 4 meters away, you have this blue spot. This is a point of discharge, and that's undersaturated. That's below saturation level of 1. So you have a very steep saturation gradient. Then if you look at what lives right here at this 4 meters where the water is discharging, you can see a coral, and there's near the 5 to 6 meters over here, another coral, and you can see here the opening of that little cave where water is discharging right at the edge of that spring or little hole. If you swim in there, you could go all the way into the cenotes and inland. Scary, but you can. I wouldn't recommend anyone do that. And you can see the corals over here. So obviously, in this system, we can find corals that are growing in these not only saturation of 2.5, but undersaturated conditions with respect to aragonite. That's very, very surprising and very much unexpected. What does that mean? That all corals can grow? What else is happening there? So, it's not all good news. The good news is, yes, some corals seem to be able to calcify in undersaturated conditions, and these specific corals spend all their life under these conditions. So as larvae, they settled over there, they divided, they grew, and calcified throughout their lifespan. But if you look at the number of species, this is the saturation above 2.5, this is 1 to 2.5, and this is less than 1. You can see that in the lagoon, outside these springs, there's 10 different calcifying coral species. When you drop to below 2.5, there's only 3 species that can make it. The coral area drops, and the number of individual colonies for some of these species also drops. So the biodiversity, even though there are certain species that can calcify, the biodiversity does decline considerably. Unfortunately, these specific 3 coral species, which are Siderastria radians, Porites astraeoides, and Porites porites, they all are not, in this specific reef systems, are not the major corals that build the reef. Yet they can grow there pretty well. The other thing we did is we wanted to look at the growth rate or calcification rate of these corals. So we took coral cores, and we wanted to look at their growth rate. We did that by cutting them and doing some X-rays and CT scans, and we looked at their extension rate, or how many millimeters a year they grow for a coral at the ojo and a coral at the control site. And you can see here the extension rate for above 2.5 and between 1.5 and 2.5 seem to be very similar, yet after when it's less than 1.5, it drops pretty rapidly. So extension rate does change, but only when you drop the pH quite a bit. On the other hand, the density, and now it's the amount of carbonate per centimeter cube of growth, or centimeter square of a cube of carbonate, does drop significantly when you go there. So while they're growing, and they're growing maybe even at the same rate, they're putting less material per unit time. And therefore, if you multiply the density by the extension, this is the growth rates, or grams per centimeter per year, you can look at and see that there is a decline in their growth rate. So while they are growing, they're not doing it as fast as the same species under otherwise identical conditions, 4 meters apart, in the same lagoon. If you look at them again, so this is from a control site, and this is from the spring. This one's from the control site, and this is from the spring. We did the CT scan, which allowed us to calculate the voids, or how much a volume of this coral is bored. You can see that as you go down to 2.5, the boring increases rapidly, meaning that potentially because their density is lower, so they put less carbonate per unit area, it's easier for boring organisms to go and eat them up and dissolve them. And the implications is that coral reefs that are built from these less dense corals that have so many borings are less robust structures. And if you have a very big hurricanes like they do down there in Mexico every once in a while, you may be destroying the reef so more rapidly. So the biodiversity decreases and the structure of the reef might be compromised and will be less robust. So it's not very good news. However, there is a bit of hope here because certain corals seem to be able to calcify and grow, and maybe we can do something to help the corals do better under these conditions. And we also looked at other proxies or other signatures of health and mechanisms to fight stress, which is tissue thickness, and we didn't see any difference in tissue thickness. This is from the springs, and this is from the control. You can even see, that's the same magnification, you can see that there is more porous structures over here. But the tissue thickness was the same. The symbionts, there's one theory that the symbionts allow corals to adapt, at least for temperature stress. It looks like when you expose corals to temperature stress, they change the symbionts that they interact with into another clade of symbionts that helps them to adapt. Here we did not see a difference in the symbionts. They were the same clade. And another potential hypothesis is that if it's harder for them to calcify, if they have more energy, they can overcome this hardship. So they just have more energy. They spend more energy pumping CO2, carbonate ions and pumping out other ions in order to balance. And then if they have enough food, they can do that more efficiently and overcome the barriers. One of the ideas is if they have more symbionts, the symbionts can reallocate more photosynthate food to the corals to help them battle this and provide them more energy. We don't have a large enough sample, but based on the number of corals we looked at, it didn't seem that there was any statistic. There was a small difference in the density, but it wasn't statistically significant. So there were more symbionts in the low pH ones, but it wasn't statistically different. The nutrient levels, however, are these submarine springs bring a lot of nutrients from land. So while the corals themselves don't utilize these nutrients, they are available for their symbionts. They're available for lots of bacteria that form biofilms that the corals might be able to eat and graze on. So it could be that the energy story is still happening. We need to follow with more work. But what we are focusing on now is trying to look at their acclimation and adaptation potential. And the way we're doing that is through transplantation experiments. Basically what we're doing is we're taking the corals, the same species, we're taking 3 cores out of a coral head, moving them one back to where it came from, another subsample from that same coral head into the springs, and the third one into the forereef out of the lagoon. And we're taking such coral heads from springs, from control sites, and from the lagoon. So a 3-way exchange with all of the combinations, and then we transplant them back into the springs, into the controls, and into the reef from the reef, we also take corals that don't even live in the lagoon, so other than these 3 corals. So we have 3 species, 2 from that are abundant at this ojos, 1 that are not found there, and we did all the transplantation. Before we transplanted them, we put them in alizarin red to mark where they were before transplanting so we can look at what grew after the transplantation. We took from each of these colonies a little tiny chip to preserve that in RNA later to do the genetic sequencing of that. And then after 1 year, we took another little subsample from each one of them to look at the gene expression. And in 2 years, which was going to be this coming January, we're going to take yet 1 more sample and then take all of the experiment out, and we're going to look at their growth rate, where we've been monitoring them so we know when they survive, when they die and who died when and so on. Every week our Mexican collaborators go and dive and check on them. And we're going to compare the gene expression for certain calcification, target genes for calcification and other, the whole transcriptome to see what's being upregulated or downregulated and what are the roles of that. And with all of these species, these 3 manipulations, we can answer A, can all of these 3 species do it? Do they do it in the same way? Is this something unique for certain species? And what are these genes that may help them adapt? And are these genes present in all corals generally, or they're unique for these species? And we're going to see how their growth rate is, if it's being impacted or not. So that's all ongoing research, and hopefully my next talk here I'll be able to present these results. What I do have now is we do know that there wasn't mass mortality of any of the species. So after the first month, there wasn't there were a few casualties, but nothing systematic that shows, oh, there's certain species all died at a certain location. And most of them, we have more than 70% survival. So, and it wasn't systematic again. So we'll have enough data to hopefully have statistically significant results, because there's about there's 90 coral heads at each one of our locations. So there is even if we only get 70% of them, there's enough replication to be able to say something meaningful, we hope. The other thing we're doing is we put these recruitment plates to see how what is recruited and how that changes over time. So we harvested and looked at what settled there after 3 months, 6 months, and 14 months. And that's it. So I just wanted to remind everybody that we need to protect the ocean. And even though I'm potentially coming with some good news that not all of the corals are going to disappear, that doesn't mean that we need to do nothing about about CO2 emissions, because ultimately, really, what the only mitigation solution is to reduce our carbon footprint. Thank you. Now I'll excuse Ivano, who has to pick up his door. And we can ask questions. By the way, I have to thank my group. That's Halloween. That's really cute. That's me, Cruella de Vil. 101 Dalmatians. No, they're not captured.
Questions?
I was wondering if you think that maybe the morphology of the coral may play a role in how they adapt, like branchy coral versus flat coral versus kind of the big head coral that you seem to be seeing at these.
That's a very good question. So the 3 coral species that we found at the springs are not branchy corals. They're more flat
and we did put a branchy coral in, so we'll see.
Cool. Thank you.
Hopefully we'll know better. The one thing I think we need to make sure is, even if, I think, which I failed to mention, is if we really want to preserve these corals, and if there are certain refugia or certain areas where the corals might be doing better, we need to make sure we protect these areas from other stressors. So one of the things with Mexico, maybe, there's corals that are adapted to living in a low pH, but then if we start overfishing and dumping sewage over there, they may get impacted by other stressors. So we need to consider that.
Adina, is that great talk? Have you measured or tried to measure the semiotics? Is just shining a ham-style porometer on them in and out of
No.
pH 8.2?
No, I didn't do that. I just extracted them, concentrated them, enumerated them, and looked at their species. But I did not do any in situ count. I don't have an in situ count. But that would be nice to do.
And I wanted to get it straight. When you showed the graphic, you called complicated, and 12 meters is not a very long distance.
Not at all.
So those graphs of the saturation level for aragonite were the permanent mean record? Or was it the,
That was
on
the
the 5 minutes it took me to take the samples, that's what it looked like right then?
No, so when we go down to the field, we collect lots of samples every few hours along these various transits. However, our collaborators in Mexico go weekly and collect samples for us as well, and strip them in little glass borosilicate tubes and reanalyze them. The calculation of the aragonite saturation was based on the average for that area, and from when we started monitoring until the paper was published. Now we have even more data. We actually have hundreds and hundreds of numbers that sometimes someone will write up and discuss actually beyond the calcification. There's a whole other story about carbonate dynamics in the black reef there, which you could calculate net calcification or respiration and photosynthesis, because we really have the pH, temperature, and salinity. We have data for 3 years on an hourly basis, which is tons of numbers. And then the discrete samples are almost weekly. Sometimes they can't go out because the weather doesn't permit, but at least monthly for 3 years.
So it's like a micro scale
It is because
chemical
of, it is because of the nature of the water. So because the water has slightly lower salinity, so the salinity in the background is about 36, 37. The salinity of the springs is about 30. So that's a big difference in salinity, although 30 is not a big issue for corals. It's enough that the water that comes out is so buoyant that it just goes up. It doesn't really spread too much. So it is really a focus, like fluid. Yes, if there is a storm, it does get mixed more, but it is pretty close.
I was curious if there are any differences in the other members of the community in springs versus the other areas, in seaweeds,
Yeah.
soft corals, other.
So the most obvious thing that when you go there is all of this biofilm. There's a lot of bacteria and a lot of biofilm covering that area next to the springs. I have another student who's doing her PhD on coralline algae. There are differences in the type of coralline algae and the relative abundance of corallines versus non-corallines. So there's less corallines, but there are there. So that's even more surprising. These are Lomapis calcite. They're not supposed to be there. They're supposed to even be more sensitive than the corals. But I guess the CO over there, it's kind of yes, they need the carbonate, but they also need CO2 because they need to
oxygen from the system, that oxygen will be different. In your system, oxygen will be different
It
because
is.
CO2.
Actually, it is.
Okay, good. I didn't see that measure, but that's important. Temperature, what we're seeing along our coast is that corrosive fluids or coincident